So I will be discussing the endogenous and the exogenous pathways of antigen presentation. And I would like to begin with the endogenous pathway. And the first step in this is that an endogenous antigen is going to be degraded by a cytosol proteolytic protein known as proteasome. And so in normal intracellular proteins are marked for degradation by a protein known as ubiquitin. And this ubiquitin protein complex will then go through this proteasome that's going to have a 20S base and 19S regulatory end. And it's going to go through there, and that proteasome complex is going to cleave the peptide bonds in that protein complex, in an ATP-dependent process. And so in our immune systems, they basically utilize this same general pathway of protein degradation. But in addition to that standard 20S proteasome that's actually found in all our cells, there's a distinct proteasome that actually appears in antigen presenting cells, and that is known as immunoproteasome. And so basically what happens is in this immunoproteasome, there are several proteins that will be replaced from that constitutive proteasome, and they're actually going to be replaced by proteins that are more specific to the immunoproteasome, and these proteins are encoded by something known as LNP genes. And I actually have a quick little figure to show you on that. Yeah, so here's exactly what I showed you. You got your proteins that will be degraded, and they're marked by ubiquitin for protein degradation. They're broken down via this proteasome with a 20S base and a 19S regulator into smaller peptides. And over here we have, you can see that beta 1, beta 2, and beta 5, and the constitutive proteasome will be replaced by, clearly you can see they're replaced by proteins way more specific to the immunoproteasome, and these proteins are encoded by the LNP genes. And what's important to point out is this immunoproteasome is going to have increased proteolytic efficiency for peptides that will be recognized by class 1 MHC molecules. So now on from that, the next step is that those peptides that were created by the protein degradation via the proteasomes in step 1, all that occurred in the cytosol, those peptides now have to be transported to the rough endoplasmic reticulum. And that's actually going to happen via a protein known as TAP. And TAP has two components, the TAP1 protein and TAP2 protein, and they will help transport those peptides into the rough ER using the hydrolysis of ATP. And I have a little something to show you on that too. So, just like I said, you can see the two components, TAP1 and TAP2, and you can see how the proteins that were degraded by the proteasome right here and formed peptides. Now that TAP, the transporter associated with antigen processing, is going to help transport those peptides from the cytosol to the rough ER, and that's all going to happen via, ATP hydrolysis into ATP and inorganic phosphate. And something cool, just a little side note, that I found out about TAP and just TAP diseases was something known as TAP deficiencies, which can actually lead to a disease syndrome that has aspects of both the autoimmunity disease and immunodeficiencies disease. And specifically, I researched something known as BLS, the Bayer lymphocyte syndrome, and this is actually a type 1 BLS because it is associated with class 1 MHC molecules, and people with this disease actually were found to have defects in their TAP1 and TAP2 proteins, or TAP1 or TAP2 genes. And so because of the defects in that, clearly TAP proteins are necessary for the loading of peptides onto class 1 molecules. So people with this disease actually showed significantly low levels of class 1 molecules, as well as being associated, or as well as showing increased levels of natural killer cells, which was very interesting. But this disease is actually a newer disease found within the past 20 years, and it actually has some pretty serious side effects, and including frequent bacterial infection of the upper respiratory tract, experiencing chronic infection of the lungs. Your bronchial tubes can be dilated so much that your lungs will be infected so much it may cause fatal damage. And one significant characteristic mark of BLS is skin lesions on your midface and your extremities. And excuse me. And the best treatment that they found right now for this is appears to be a combination of antibiotics and just injections of immunoglobulin. But the problem they actually found upon more research is that the problem was in the promoter region of this TAP gene. So they were thinking maybe gene therapy would be best, but just because the cellular distribution of class 1 is so widespread, that's really not clear what cells they'd have to target correctly to alleviate all these symptoms. So that's just a little side note of a disease that I found that was very interesting, and it all related to TAP deficiencies. So after our peptide's been transported to the rough endoplasmic reticulum via that TAP, basically these molecules known as chaperones are going to aid these peptides to be assembled with the MHC class 1 molecules. And so how's this going to happen? I got one more diagram for you. Here, you'll see our first chaperone I would like to talk about, calnexin. Calnexin is a chaperone that is going to associate with the free class alpha or free class 1 alpha chain, and it's also going to be associated with this protein ERP57, and it's going to promote the folding of that alpha chain. Then you're going to have your beta 2 microglobulin that's going to come in and bind to the alpha chain. Can you see this? Yeah. It's going to bind to the alpha chain. The calnexin, that first chaperone, is then going to be released, dissociated. And you're going to have two new chaperones that are going to come in. Calreticulin and tapacin. And that tapacin is also basically going to do, it's going to bring that TAP transporter into closer proximity with that class 1 molecule. And that closeness is going to allow it to acquire an antigenic peptide. As you can see, eventually the peptide and that class 1 MHC molecule will be together in a complex, and at the very end, due to the increased stability that was due to the productive peptide bonding, this class 1 molecule can then release and actually allow these chaperones and that protein to dissociate, and that class 1 MHC peptide will eventually be transported from that rough endoplasmic reticulum to the Golgi complex to eventually the plasma membrane, as you can see the complex leaving the rough ER there. So yeah, that's basically a summary of the endogenous pathways, ending with those chaperones helping to aid in that peptide assembly with that MHC class 1 molecule. And then that MHC class 1 molecule and peptide complex can then exit the rough endoplasmic reticulum and make its way onto the plasma membrane. And so then I would like to talk about the exogenous pathway of antigen presentation. So to begin with, our antigen presenting cells actually express both class 1 and class 2 MHC molecules. So because this is a thing, excuse me, there has to be some mechanism, some way that's going to prevent these class 2 MHC molecules from binding to the antigenic peptides that are destined for the class 1 molecules. So what's going to help recognize the difference? It's something called the invariant chain. Basically, this invariant chain will interact with the class 2 peptide binding groove, and it's going to prevent any endogenously derived peptides from binding while the class 2 molecule is within the ER. So basically that invariant chain is going to say, hey, and no endogenous activity. It's going to send it around the right pathway to exogenous activity. And that's going to be happening because that class 2 MHC molecule, its alpha and beta chains will associate with the invariant chain. And so further, the invariant chain is going to help because it's going to contain sorting signals in its cytoplasmic tail that will direct the transport of these class 2 MHC molecules through the trans-Golgi network to the endocytic compartments. So basically the invariant chain has, one, helped us in deciding or helped us in preventing any class 1 MHC molecule antigenic activity from going on, thus sending us down that class 2 exogenous pathway. Second, the invariant chain has helped transport through the Golgi apparatus to the endocytic pathway compartments. And now the invariant chain is eventually going to become degraded. But it's going to leave behind something known as the clip fragment. The clip fragment, class 2 associated invariant chain peptide, and it's going to remain bound to that class 2 molecule even after a majority of the invariant chain will be cleaved. What this clip fragment will do is going to physically occupy the peptide binding grooves of that class 2 MHC molecule, and it's going to prevent any premature binding of antigen-derived peptides. So after this clip fragment is left behind, eventually our exogenous antigens will be taken up, degraded, and routed to endocytic pathway compartments. And so now something is going to come in that's going to mediate the exchange of the clip fragment for an antigenic peptide. Because eventually we want this antigenic peptide to pair with a class 2 MHC molecule and be transported to the plasma membrane. So what is our mediator? Well, our mediator is something known as the HLA-DM. And what it's going to do is associate with class 2 MHC beta chains, and it's going to function in removing or editing peptides that associate transiently with the binding groove of classical class 2 molecules. And so I have another diagram to show you real quick of how this HLA-DM molecule is working. So basically, as I kind of already told you, we got the alpha and beta chains of the MHC class 2, and that invariant chain will come in and help us go down the exogenous pathway instead of that endogenous pathway. And that invariant chain is going to help transport it through to the endocytic compartments. And eventually that invariant chain will be degraded, as you can see, digested invariant chain. But what's going to be left behind is the clip. And that clip was physically occupying that peptide binding groove, preventing any premature binding of antigen-derived protein. And here's the HLDM that I was talking about mediating the exchange of this clip for the antigenic peptide. And one thing to point out is those peptides that are making especially strong molecular interactions with the class 2 MHC, those really, really strong bonds are what's going to be super hard for the HLA-DM to come in and displace. And thus those peptides with those strong interactions are really what are going to be ultimately make it to the very end, to that cell surface as an MHC peptide complex. And so to end with, you're going to see that class 2 MHC peptide is transported at the very end to the plasma membrane. So basically once a peptide has been bound, the class 2 MHC peptide complex is transported to the plasma membrane, where that neutral pH of the plasma membrane is going to appear to enable the complex to assume a compact stable form. And I'm going to put my laptop down, but the last thing I want to talk about is this. What is this? This HLA-DO right there. Well, what that is it's basically another non-classical member of that class 2 MHC family. The HLA-DM is actually another non-classical member. But this HLA-DO has actually been found to be a negative regulator of antigen binding, and it's basically modulating the HLA-DM the whole time, and basically changing the class of peptides that are actually going to prefer to bind to that classical class 2 mediator. So, yep, that's pretty much the end of the exogenous pathway, ending with class 2 MHC peptide complexes being transported to the plasma membrane. So there's your summary of endogenous and exogenous pathways of antigen presentation, and feel free to use my video